Good afternoon and welcome to the Medical Center Hour. This is a program entitled The Malaria Project, the Secret Government Mission for a Cure. I'm Marcia Day Childress from the Center for Biomedical Ethics and Humanities. Happy to welcome you here today to a program that is also one of our History of the Health Sciences lectures. During World War II, with malaria a real threat to American troops fighting fitness, especially in the Pacific Theater, the U.S. government mounted an all-out hunt for a malaria cure. The story of this endeavor is a very complicated one, with many threads and many angles that give one ethical pause. It's also a story that went untold and unexamined for several decades, and a story largely unknown still, even among experts and specialists in infectious and tropical diseases. While learning to mine the U.S. National Archives, public health journalist Karen Masterson happened upon archived documents that had been incorrectly boxed. What she found there put her onto this story of secret science in the service of America's war effort, experimentation in pursuit of an anti-malaria drug that took its cues from early 20th century German research models and methods, and that involved risky research on institutionalized and incarcerated Americans. We're pleased to welcome Karen Masterson, who's a lecturer at Johns Hopkins University in the Writing Seminars and Public Health Studies programs. In this History of the Health Sciences lecture, she'll tell us the astonishing, sometimes appalling tale. This episode in the history of recent science helps us to appreciate the complexity of research, how science and medicine are always part and parcel of their social, cultural, and political contexts, how federal rules and regulations governing ethical human participation in biomedical research weren't always in place. And indeed, if they had been, much of this malaria project could not have been done. It's also instructive to recognize the thorny business of weighing competing risks and priorities, whether we're exercising hindsight or thinking about our own difficult choices here and now. Following Ms. Masterson's talk, UVA Professor Emeritus of Medicine, Dick Pearson, will offer a brief response from his perspective as a longtime infectious disease and tropical medicine specialist and very interested reader of Karen Masterson's book. Her book, The Malaria Project, is available for sale and signing by the author just outside the auditorium. Again, we'd like to thank the History of the Health Sciences lecture series, a program of historical collections here in the Claude Moore Health Sciences Library. And now, Karen Masterson and then Dick Pearson. Welcome.
Thank you, Marcia, and thank you everyone for being here today to listen to the story of the Malaria Project. I became interested in this story because I stumbled onto the bioethics issues. But what really drew me in and kept me interested was this fascinating disease. I'm not a bioethicist. I didn't know much about bioethics. I didn't know very much about malaria or World War II before I found the story. I was actually a political reporter. I worked in the Senate press gallery in the Capitol. I chased down senators and members of the House. I interviewed Hillary Clinton when she was a senator probably a half dozen times. And I wrote stories about legislation and policy and politics for a big Texas newspaper. But I left that world for a more sane work-life balance. As a science writer, before I became a political reporter, I was an environmental reporter for the Philadelphia Inquirer. And so my first point of transition was to accept a fellowship at Johns Hopkins, where I could study the history of medicine and infectious diseases, and it was there that I really did the bulk of my research for this book. I was in the National Archives looking for something completely different when I stumbled onto a memo from the National Research Council to the lead health official in the state of Massachusetts asking if researchers could come in and infect Boston psychopathic patients with malaria so that drugs could be tested on their infections no less than the war's outcome was at stake. Now, I was looking for records online appalling, and I found this. And I had no idea what to do with it. So I just kept pulling threads, and I worked with archivists because the process for finding records is convoluted, and this wonderful archivist that I work with found all kinds of records for me, and after about 500 boxes I had pieced together what I called, and what casually was called back then, the Malaria Project. It was really, it's what Marcia said it was the US government's response to this problem that they had during World War II. Troops were falling first by the thousands, then the tens of thousands, then the hundreds of thousands. Over 500,000 US troops were infected with malaria. That's just US troops. Malaria was a problem for both sides of the war. All countries who had committed troops to battle had to deal with this problem because we were fighting in areas where transmission rates were up to 80%. So the War Department asked scientists to help. This is the box where I found that initial memo when I was looking for something online was calling. I found a lot of records that looked like this. They were top secret. This was a secret project. And it was a secret project because everybody knew that a malaria drug would be a weapon. It would be a weapon that would give your side an advantage over the other, because nobody had anything good. Quinine was the only known drug. It had been blockaded. It was a drug made from cinchona tree bark, and the cinchona tree plantations were all in Java, and the Japanese had blockaded these plantations. So quinine, they didn't know how to use it, they didn't know what to do with it. The Dutch owned plantations, but they blockaded, so nobody had access to the quinine. Hundreds of reports that look like this were written by lead investigators for the American project, and they enumerate the number of bodies used and what happened to those bodies. So I could run a tally in my head as I was going through all these reports of how many people were used, and they were prisoners, they were state hospital patients, they were POWs, they were conscientious objectors, and they were soldiers. To understand the bioethics of this project, though, I feel it's important to give you all the context too. So if you'll bear with me, I want to give you the backdrop, what was happening all around, what happened before this project began. So when I get to the use of these patients, you have a full understanding of why scientists crossed that line, and they seem to do it so easily. It all started with this man, Julius Wagner Jahre. He was an Austrian psychiatrist who has a wonderful backstory that I include in the book, but for the purpose of this talking, because I always talk too long and I want to make sure I stay within my 30 minutes, suffice it to say that he had figured out that a certain subset of psychiatric patients who had neurosyphilis, the last stage of syphilis, when the spirochetes actually got into the brain and caused damage, that category of mental health patient could sometimes be cured if you gave them malaria and allowed the fevers to escalate to their peak a few times. And he understood it back then. He wasn't even a malariologist, but he understood it back then kind of intuitively that it was boosting the immune system to fight the infection of the syphilis infection that actually helped kick it out of the system, kick it out of the brain. And so he saw, after giving malaria to over 1,000 patients, that if he caught those last stage syphilis patients coming in to the state hospital system before they were too far gone, that he could actually cure them about 30% of the time. The British saw this and thought it was brilliant. Yarg had these strictures. You only use Vivax malaria. Vivax is the kind of malaria that you get in Asia, back then in southern Europe, in the southern United States, in South America. Vivax is the kind of malaria that will hibernate in your liver and launch relapses, but it's rarely deadly, versus African falciparum malaria, which is very deadly, and you could start showing symptoms in the morning and be dead by the end of the day. So he said, no, you only use Vivax malaria, and you only use blood transfusions. You don't transfuse the disease through mosquitoes, because if you did that, then you would be curing syphilis patients of malaria, I'm sorry, syphilis patients of the syphilis, but you'd be sending them out into the community with the malaria still in their liver. So he had these rules, and he stuck by them. Here you can see a very sick patient in the background with malaria at the height of the infection, and they took blood from him and they're injecting it into this patient in the foreground to give malaria to that patient. He had to keep his strains alive in people because there was no way to culture the parasites. So he always had some of his patients infected with malaria, and he could collect his data, and he collected his strong ideals on how to use this. The British tested his theories at their state hospitals, and they found they did, it's called malaria therapy. They gave malaria to 1,600 paretics. The late-stage syphilis patients back then were called paretics. And of them, 36% died of the malaria during the malaria infection. 25% walked away fully cured, and the rest remained unchanged. So 36% died, 25% cured, and that was considered a success. Right? They had a control hospital where they had about 100 patients they were tracking, and within 2 years, all but 3 or 4 of them died. I'm forgetting the exact numbers, but most of them died within 2 years. So Yard won the Nobel Prize because he figured out how to treat a mental illness, and he's only one of 2 psychiatrists in history to win that prize. The Germans capitalized on this idea. Bayer and Company, which was part of the IG Farben conglomerate, went into the state hospitals and said, can we use your malaria therapy patients to test our experimental drugs on them? We will vet them on animals first to make sure that they're not too toxic, but we just want to see if our drugs will work against the parasites. And you've got lots of malaria here in your state hospital. Will you work with us? And they said yes. So the Bayer and Company wanted to displace the Dutch in the malaria market. They wanted to create a synthetic version of quinine, put those slow-growing trees and that expensive process of extracting quinine from the bark out of business, and have another blockbuster like their Bayer aspirin. Because back then, malaria was everywhere, and people took quinine like aspirin. And so they wanted that market, and they had the goods. They were far ahead of all the chemists in the world on creating synthetic drugs, and so they actually had some leads that they got from Paul Ehrlich, actually, on where to start, and they ended up making 2 drugs, Adabrin and Plasmachin. Plasmachin was the first one. They made that around the time that Yard won his Nobel Prize, and in the state hospitals it looked like it was quite active against the parasites, but once they did bigger studies in Eastern Europe on larger populations, they learned that actually this drug actually destroyed blood cells and was a real problem. And so they kept working at it, and in the early 1930s they came up with Adabrin, which was based on a yellow dye. And Adabrin its toxic effects weren't that bad, they weren't deadly, but it wasn't a great drug, and it didn't displace quinine. It was mostly used in the German markets in South America, which is why this is in Spanish. And it didn't really get very far. Well, fast forward to 1930s Germany, late 1930s Germany, and Hitler's plan to prepare for war. IG Farben, this is their headquarters in Frankfurt, had been co-opted into the Nazi system, and their orders to Bayer were to find a better drug that that German troops needed to survive these infections against malaria that they knew would be a problem in the places where they planned to do battle. And so Bayer got to work, and one of the scientists who was pulled into the process was Klaus Schilling, who in the 1920s and 30s was probably the leading expert in malaria. He did really innovative things. He milked the salivary glands of mosquitoes to get the saliva, and he would inject it into patients and then give the patients aspirin to suppress the fever, so he could continue giving the patients that first stage of malaria. To understand this, that malaria has several stages. I talk about three of them: the infant, the adolescent, and the adult. Dr. Pearson can talk about the technical side, but as a storyteller, these are the three most important versions. And the infant version is when they're in the saliva of the mosquito. The mosquito bites you, gets into your body, immediately goes into your liver, and one parasite turns into about 40,000 or 30,000. And in a week or two, they burst out of your liver and they attack your blood cells. And inside your blood cell, one parasite will turn into 30 or 40. The cell will burst, and out pours another whole army of these parasites, so that within a week or so, you've got trillions of them in your body. And at a certain stage, some of them mature into the sexual stage, the adult stage, and they circulate into your surface cells, emit a chemical that makes you 100% more attracted to mosquitoes. This is actually a mosquito attractant. And the mosquitoes come and bite you, drink in that adult stage, and in the mosquito's gut, they fuse into an oocyst, or an egg sac, and a week or two later, based on air temperature, those sacs burst with these new infant versions, these stick-like versions in the saliva. And so what Schilling wanted to do was boost the immune system against that first version of the parasite, so that your immune system would recognize it quickly and stop the infection. Never get into your liver, you'd never have an infection. And he was actually making some progress, but war started, and so he had to go to Heinrich Himmler and ask for an opportunity to study this. And Himmler said, sure, I'll give you the Dachau concentration camp. And so Schilling was sent there, and he infected about 1,000 prisoners with malaria. They actually, in his way to the hospital, they had clean sheets. They were given an extra ration of food. They had to be relatively healthy, otherwise he couldn't take them. You can't give malaria therapy to people who are already dying. And so, from some perspectives, his patients actually lived longer being in his hospital ward as part of his studies than the inmates who were exposed to typhus and the SS cruelty. He stayed when the Americans liberated the concentration camps. He stayed. The SS all fled, right? He stayed to tell the JAGs, the army lawyers who were overseeing the hearings, that he had this great data, that he had studied malaria, that he had found some great results, and that his colleagues who looked up to him before the war would want the data. And here he is trying to explain, yes, I infected inmates and I kept them sick and their fevers went, yes. But you don't understand, this is what we do. We're malariologists, this is a part of what we do. The army lawyers didn't know anything about YARG or malaria therapy or the direction that all this research was going in. And so he was convicted and he hanged, he was hanged. And that pretty much was the end of Germany's lead in malaria research. On the American side, things went very differently. This is Lowell Coggeshall in the suit amid all the white coats. After the war, he became the dean of the University of Chicago's medical school, became the vice president of the university. He advised President Eisenhower on medical policy, especially medical education policy. He said, we are focusing on body parts too much, that specialties are the wrong direction to go, and we're pumping out way too many students who are specialists and not enough who understand the whole body. We should be working with nurses associations and public health associations. This is a whole body approach. And in the 1950s, he was called a communist in Washington, and so he retreated back to the University of Chicago and continued his good work. But I found him a relief. So I found what I thought was a man who was a good man. He was actually a nice man who was the lead thinker on the Americans, creating this model based on the Germans' work. And so I use him to tell my story, and I start with when he was just a cub graduate student in the swamps of Leesburg, Georgia, studying malaria for the Rockefeller Foundation. The Rockefeller Foundation then was what the Gates Foundation is today. They invested $100 million into getting rid of malaria in the U.S. South, which is about $1.5 billion in today's dollars. So they were a big part of it, and he was very fortunate through an interesting guy, came from nothing, was a poor farm boy, and he wanted to get out of farming, so he decided to study biology, and he ended up through a series of good luck and hard work, ending up with the Rockefeller Foundation, and they turned him into a malaria expert so that by the time the war started, he was the lead thinker on how to tackle this problem before we committed any troops to the ground. Here he is in one of the first meetings of the Malaria Project, talking to other experts about how they were going to convince the War Department that the War Department needed this. Well, the War Department wasn't ready to listen, and as they were formulating their ideas, Coggeshall got pulled away from Washington because the U.S. was secretly arming and supplying the British, who had been cornered in Egypt by Rommel's forces. And the way we were doing it was we were running Pan Am Airlines ran they were the front for the Army Air Command, running supplies from Miami down to the elbow of Brazil, across to the hump of Africa, and then across the Sahara into Egypt to help Brits. And everything was stopped right there at the hump of Africa because of malaria. Everything, and the Gold Coast was what Ghana was called back then, and Liberia, where our airstrips were nearly 100% infection rate for malaria. So Juan Tripp, the head of Pan Am, begged Coggeshall to come and help, and he did. So we went overseas, and this is what he found. He found the jungles just torn up to move supplies and to build airstrips. So when you tear up the landscape, you create more breeding grounds for mosquitoes. And so what he saw was a proliferation of mosquitoes, and he quickly realized, wait a minute, our problem might not be just the drugs, our problem might be mosquitoes too. So he said, we're going to screen everything. The British Royal Air Force said, we've been here forever, back off, we know how to deal with malaria, screens aren't going to do it, you need the quinine. And so Coggeshall said, well, all right, let's create a study. So he had 100 Royal Air Force pilots on one campsite and 67 Pan Am pilots on the other campsite. He wrapped the Pan Am folks in screen. All the barracks were screened. They had to use bed nets if they weren't in a screened area. They had to wear their clothes rolled down, their pants rolled down, their sleeves rolled down. So that was a very strict control of their behaviors. And then the British just had quinine, no screens. And of the 100 British pilots, 87 of them came down with malaria, losing over 1,000 man days. And of the 67 that were wrapped in screens, only one came down with malaria, and it was because he snuck out for sex. And so what Coggeshall figured out was that, oh, okay, then you have to sanitize the sex villages of mosquitoes as well. There's a big backstory on Liberia and how we used that country and what transpired there while we were getting rubber from the rubber plantations and using that as an entryway to the war to the north that I don't have time to get into, but it's very fascinating. And so Coggeshall came back from the war with a more informed approach that he brought to the War Department. He said, okay, we do need the drugs, but you need to have people in those combat zones who understand malaria doing anti-mosquito work, because you can't kill all the mosquitoes. And by the way, only about 40 of them carry malaria, and they're all anophelines, so you do targeted studies and anti-mosquito work. And the War Department said, no, what, too much. We've got more than 500 camps training men to go to war. Wait in line. And that's pretty much the way it was until Bataan. When the Japanese bombed Pearl Harbor, they next went to the Philippines, and they attacked MacArthur's troops. MacArthur's troops were all forced out of Manila and down onto the Bataan Peninsula, which the Rockefeller Foundation had already studied and knew that 30% to 80% of the local people were seeded with malaria. Fortunately, his troops had a supply of quinine that lasted them about a month. And when that quinine supply ran out, they started falling. And they fell by the thousands. So that by the time of the surrender, 80% of the troops were infected with malaria. The commanders all wrote back saying, we had plenty of munitions, we had plenty of big guns and rifles, but we had nobody to man them. Everybody was sick. That woke up the War Department, and they called Coggeshall and his colleagues, and they said, we're opening the spigot. Tell us how much you need. set up whatever infrastructure you need for this. It was folded under the Office of Scientific Research and Development, which is where all the wartime science projects were contained and organized. It was a secret White House office that got funding that wasn't approved by Congress. It's where the Manhattan Project was housed. So now the project is going, and in the next months, over 400 scientists were brought into the fold, 50 universities, and most of the large pharmaceutical companies, and about a half dozen state hospitals, and to start with 3 prisons to do the clinical work. All this was getting pulled together while we were landing 10,000 Marines on Guadalcanal, ripping up the landscape. The waters pooled and created perfect mosquito habitat. The camps became mud holes. The shelling and just the total destruction of the landscape, again, produced prolific numbers of mosquitoes. And one of the medical corpsmen said to one of the battle commanders, I see a problem here because we know the local people are seeded with malaria, and now we have a lot more mosquitoes than when we first arrived, and the men are all getting eaten alive at night. We're going to have a problem with malaria, and the command, this is recorded, is sent back to DC, to the headquarters. The commander said, we're here to kill Japs, and the hell with mosquitoes. That month, the same time that report came in, the first cases of malaria started filling the makeshift hospitals. And on Guadalcanal in New Guinea, by the end of 1942, the infection rate was 3,000 per 1,000 men. So they were getting it repeatedly. The only option that the War Department had, because the malaria project was just getting underway, was just coming together, was this drug that I mentioned earlier, this yellow drug, Adibrin, that the Germans had made, and it was toxic. We didn't know how to make it properly, so it was also inconsistent. Our drug companies did their best, but they didn't do that well. And we also didn't understand the dosing, so that we actually, when the orders went out to put all troops on this drug, we overdosed them. So that by the 3rd pill, twice a week, and by that 3rd pill, reports were coming in of troops doubling over with cramps, soiling themselves with uncontrolled diarrhea, throwing up already meager rations in these battle zones. In Tunisia, historians have recorded this in some of the battles. There's a battle for Hill 609 where it was hand-to-hand combat, and our men had to stop fighting because they were soiling their pants. It was a nightmare. And then a rumor went around that it caused impotence, and that was it. The men weren't going to take it. They were not going to take this drug. The sands of Guadalcanal were made yellow by 10,000 Marines spitting out their Adibrin pill. They were supposed to take it in their mess tent. They'd step outside and they'd just spit it out, and the sands were yellow. So the War Department turned on their propaganda machine, which contained Theodore Geisel. He was in the office, and so he drew these wonderful posters, and they became ubiquitous. They were everywhere. They were out in the field. They were on the bumper stickers of all the Jeeps. They were included in the trailers of the movies. You know, I don't know if you know anything about World War II, but going to see a film was a really big deal. And so these posters and cartoons that were produced by the Looney Tunes, who after the war produced the Looney Tunes series, they created these cartoons. The troops, these were so ubiquitous that the troops started calling mosquitoes Ann for anopheline. They were slowly being indoctrinated into this notion that you had to avoid these anopheline mosquitoes if you wanted to avoid malaria. Billboards went up. They tried sex to get the attention of the men. Take Adibrin. This brief one says, Adibrin keeps me going. And then the War Department started training units, malaria units that were trained in identifying and dealing with malaria that then attached to the combat units. They were trained in, they have dipsticks, they're dipping into the pond to collect larvae so that the larva could be studied, so they could learn how to see the difference between an anopheline larva and a regular mosquito. Because this is all about, you can't kill all mosquitoes, right? There's just too many of them, so if you target the species, then you can control malaria. And so here they are learning. This is at a camp in New Orleans. This is before they shipped out. General Patton actually had his units in place after the Allied troops took Tunisia and were ready to launch the attack on Sicily and then get to Italy. And remember, this is all before Normandy. So the approach to Hitler was through his underbelly. We were North Africa, Sicily, Italy, and then up through the Alps. And Patton didn't understand these guys because they didn't carry rifles. They carried dipsticks, and he called them dipshit units. And so he left his behind in Tunisia. He didn't take them to Sicily with him. And so he didn't know, he didn't have anyone there advising him on what to do when 30% of his troop strength was eroded by sick men with fevers. He went into 2 hospitals, it's pretty famous. He went into 2 hospitals, not just one. I mean, there's one hospital that's the most famous, but he went into 2 hospitals and slapped soldiers because they were sick. And historians now look at those 2 cases and they believe that both men had malaria. Eisenhower almost fired him, but he was just not programmed to deal with malaria. It had to be forced on him, whereas MacArthur was the opposite. MacArthur totally got it. After what happened to his troops in Bataan, he was all on board. He ordered more units than the training camps could provide. He had his units hiring local men by the thousands to clear the debris out of the rivers so that the water would run, because running water kills larvae. He had them spraying tire tracks with Parish green because these islands were ripped up by these trucks and by shells. And so he also had units just studying the larvae and the mosquitoes so that and the local populations to see how seeded they were with malaria, so that targeted controls could be put in place. All the doctors had to go to malaria school so that they could learn the difference between falciparum and vivax. There's actually 4 kinds of malaria that humans get, but really the two big ones were these 2, vivax and falciparum. They had to understand the difference between a relapse and a recrudescence. And they had to understand what blackwater fever was, which was an overuse of quinine, and then the infection causing black blood in the urine, and it usually led to really uncomfortable death. MacArthur, as you can see here on the right, even went to school. This is in Australia. He really wanted to learn about malaria. There's an interesting backstory about a Captain Eggeberg who was in New Guinea trying to tell the services of supply that he had no BD rate. They kept asking him, SOS kept saying, send us your BD rates. And he would say, I don't have BD rates. I don't have venereal disease. We don't even have any women here. We have malaria. Please send me malaria supplies. And they would write back and say, you're about to be disciplined because you haven't sent us your BD rates. So he sent back a rather colorful letter using an F-bomb to say that they had sheep, and if they wanted him to investigate sex with sheep, he would do it. But there were no women, and so the MPs were sent out to arrest him, and MacArthur's malaria experts stepped in. They did a study, and they just provided the data that the SOS command needed to recognize that it was a malaria problem. And MacArthur, to his credit, then so admired this guy for being direct, hired him as his personal physician. MacArthur was on it. The Air Command, the Army Air Command, jumped in. This is Italy. I'm jumping forward a little bit. When the Germans started to retreat from Italy, they destroyed all the dikes and pump systems that had kept the countryside from being flooded, knowing that the flooding would create anopheline mosquitoes. And so when the Fifth Army and the British forces were coming through in May, it would be about malaria season, and they expected that it would slow them down. So this was it was an act of germ warfare. This was all going on overseas while the malaria project back home was coming together. James Shannon, who was the future NIH director, ended up being the head of the clinical studies. He worked what was then called Welfare Island, is now Roosevelt Island. he had written the state officials to ask that all of the appropriately healthy mental health patients throughout the state be collected here on this island at Goldwater State Hospital and Manhattan State Hospital so they could be used, so they could be given malaria and used in these studies. Now we're not talking about paretics anymore. We're not talking about just neurosyphilitics. We're talking about schizophrenics, people with bipolar disorder. Anybody who was healthy enough to withstand the malaria therapy was sent to him. And he called this was his clinical material. This was his biological thinking dialed way up. And it was the exigencies of war, and he stepped up and met the demand and figured out a way to collect as many bodies as he could. He used prisoners at Sing Sing. This is Sing Sing prison. He also used them at Rahway, New Jersey. This is a picture of Boston Psychopathic. The project got up and running there, led by Alan Butler. St. Elizabeth's Hospital in D.C. with its 6,000 beds was also used. This is G. Robert Coatney. He was a parasitologist. He wasn't even a doctor, but he led the clinical studies in the South. His prisoners were in the Atlanta Penitentiary, the U.S. Penitentiary in Atlanta, which was not a high security prison. He used state hospital patients at Milledgeville, the Milledgeville State Hospital in Georgia, at the South Carolina State Hospital, and by now, you know, it's well into 1943, and they've been at it, and they're not finding anything. They're testing thousands of compounds on bird malaria and hundreds on people. Most of them are sulfur drugs. They're not really having any breakthroughs on novel compounds. Everybody's mad at each other. The chemists who are working on bird malaria are frustrated because the clinical researchers want to take drugs to the patients far sooner than they think they should. And besides, bird malaria doesn't really extrapolate to human malaria. Your model's all wrong, and they're fighting one another. Coatney is complaining because the military keeps taking not only his lab assistants and sending them overseas, but also the inmates. So inmates are getting early release. If they're not high security inmates, they're getting early release, and so they're in the middle of a study and they're getting dragged out. As you know, because you know the leaders in this project are having little meltdowns. They have a breakthrough during the liberation of Tunis in Tunisia. A French doctor comes forward, corners an American infantryman, and says, "I have the cure for malaria. I have a drug that's made by the Germans. Bayer made this drug. They made me test it in the adobe villages of Tunisia right before you all arrived to start this battle. I collected this data on them, and I can show you that it not only cures it, but it prevents malaria. And I want you to have it because I don't like the British and French. I don't want the British to have this. I want you to have it and send it home." And it took several months. I found the report in this box, but it took several months for it to be translated from French into English. And by the time it hit the project had very few bodies left. They had so used their bodies, their state hospital patients and their prisoners, that they needed to open up a whole new wing of their effort. And this man led the effort. It's this Alf Alvey. He was a University of Chicago professor, Swedish. He was a renal expert, and he reluctantly, almost by force, took over a triangle effort that includes the University of Chicago, Manteno State Hospital, which is about 30 miles south of Chicago, and then 20 miles to the west. This is where he grew the malaria. So by now, they're using the state hospital patients to grow the parasites. They're taking parasites from troops from both sides of the war. They're giving falciparum, Vivax, different strains of both, Ovalia if they have it, Malaria, the two other, there's anything that they're taking from the soldiers' blood, they're injecting into these state hospital patients and growing the parasites there and then driving them to the prisons. This is Stateville Prison, which was what Alf used. Now, Alf was innovative because he decided that he wasn't going to worry about, this is a high security prison, so they're not going to be released. The inmates aren't going to be released. And he decided he was going to use the smartest ones there. He was going to canvass the guards and the warden and figure out who the smartest inmates were, and he was going to use them as his lab technicians. And his most productive was Nathan Leopold, who was looking straight at you in the middle of the photo. Does anybody remember the Leopold and Loeb murder case from 1924? Richard Loeb, Nathan Leopold, brilliant, rich sons of business titans in Chicago, picked up a neighbor off their mansion-lined street and killed him just to see if they could get away with it. And then they helped the journalists and the police with their investigation because they were so confident that they were so smart that they were going to just get away with this murder. And they weren't that smart, and they did get caught. And so this is Clarence Darrow in the foreground, who came up with a very clever precedent-setting in the annals of criminal lawyer, criminal law, the history of criminal law. He saved them and got them 99 life plus 99 years. Richard Loeb died in a shower stall fight, but Nathan Leopold lived on to kind of run his little fiefdom at the prison. And when Alf Alving arrived there, Alving knew to use him. Alving couldn't stand him, but he knew to use him. And so he became one of the most productive lab technicians. He was even used in the experiments and then was released later because of it. He actually earned his release from prison because of this malaria work. What they made from this German compound that was captured in Tunis is the drug chloroquine, which after the war was used like aspirin in the tropics in the World Health Organization's first ever attempt at eradicating an infectious disease. And it is credited with saving more lives than any other synthetic disease made for infectious diseases. But by 1950 in Asia, resistance cropped up, and then by the 1970s, that resistance, parasitic resistance to the drug had spread throughout the world, throughout the malarious world. And that's pretty much the story of every drug that's ever been made against malaria. Most of the drugs, almost all drugs that we use today and we know of today came from this World War II project, except the artemisinin, which is an herb, a Chinese herb. And even the artemisinins now are following this pattern. So for me, we're still looking for that pill, all that work, and we're still looking for a pill. I came away from this research thinking that Coggeshall actually had it right, that drugs may not be the answer, they can be helpful, but that you need to work with the conditions on the ground. And so in the context of the 21st century, that is working on health systems first and drugs second. And the biggest lesson for me in thinking about this is that most of the money going towards malaria research now actually come back into our labs for drug and vaccine development. And a minority of it, very, very little of it when it comes to malaria research, although more, if we're talking about AIDS research, goes into building health systems on the ground. On the bioethics side of this, which is I know what you're mostly interested in, William Bynum reviewed my book for the Wall Street Journal and said, Masterson allows her readers to make up their own minds about whether war can be good for science and medicine. I came away thinking that even if it is, the price is frightfully high. So with that, I'd like to stop talking, and I know Dr. Pearson is trying to come up and he probably has, may correct me on some of the technical stuff that I perhaps got wrong.
I can't correct you.
But thank you very much.
It is a tremendous honor to participate today, and I have a number of comments. I was, let's see, oh, here we go. Got all my equipment together. I was trying to think of a notable quote that might capture a little bit of the impact of this book on me as I read it. There were many, many points of connection. But I went back to Isaac Newton. If I have seen further, it is by standing on the shoulders of giants. And I think, indeed, we've seen a lot further today through Karen's outstanding talk and her masterpiece, the Malaria Project. And I want to emphasize the mission to find a miraculous cure. Janet, my wife, is here, and the two of us in 1977 were in Monrovia, Liberia. We had landed at Roberts Field. We actually saw a picture of that, the site of an Army Air Force base constructed during World War II and plagued by malaria. I remember the frantic knock on our door late one hot, steamy evening. It was rare to have an unannounced visitor at that time of day, as Janet will tell you. And as I opened the door, I was confronted by our watchman, who is holding this limp young child, approximately of the age of 2 years. And it was immediately apparent she was very sick, and I put my hand on her, and she clearly had a high temperature. So we had an old Toyota. The battery didn't work. It was parked on top of the hill. He helped me push it down. It started, and we made it a hurried run to the John F. Kennedy Medical Center, where I was serving on the faculty. I don't have that patient's blood smear, but I brought one from the literature, and it would have looked something like this. You can see the red blood cells. This is a normal one. It has no nucleus. And here you can see malaria ring forms within those erythrocytes. Approximately 40% of these cells were infected, and falciparum malaria infects cells of all ages and can be fatal within 24 to 48 hours. At that time, in West Africa, 25 to 50% of children died before they reached the age of 5 years. Malaria was the principal cause of that death. By the year 2000, there were more than 1 million deaths annually around the world from malaria. More than 90% of those were in equatorial Africa. And of those, the vast majority were in children under 5 years. In fact, most were under 3 years. So our watchman's daughter was infected with a killer, and it was clear that her life hung in the balance. As I read the Malaria Project, I was captivated by the fascinating accounts of the giants responsible for the prevention of malaria and the development of this miraculous cure, chloroquine, in the first part of the 20th century. And they were giants indeed. They risked their lives in the swamps of Georgia to the battlefields of Guadalcanal. And as Karen has pointed out, many were flawed, some terribly so, using patients in psychiatric hospitals, prisoners in concentration camps, convicts in American prisons, without informed consent and human safeguards. But at the end, though, the objective of that project was achieved. Several days after that first knock came a second. This time our watchman was back. In one arm, he was holding his alert daughter, who had recovered from her malaria. His other arm was holding a chicken, a live chicken. Janet can tell you the rest of the chicken side of the story, but I was left with a wonderful memory of what it is to stand on the shoulders of giants and use this miraculous drug, chloroquine, to save her life. Parenthetically, it's important to add that Janet and I were taking weekly doses of chloroquine to prevent malaria, as were other expatriates, and we remained healthy throughout our 1.5 years in West Africa, which at one point was known as the white man's graveyard because of the risk of malaria to those of us who did not have immunity. So what is the current status of malaria in the year 2016? Here are the most recent data. Half of the world's population remains at risk of malaria. 214 million cases occur each year. That would be more than 90% of the American population. The bad news is that 438,000 people die each year of malaria. The good news is that's down from 1 million a year in the year 2000. Much of this relates to the rollback malaria program with 500 partners, including the World Health Organization, the US government, and the Gates Foundation, which overall has seen a 60% decrease in malaria deaths, I think in large part using the infrastructure and developing infrastructure. This is attributed to insecticide-impregnated bed nets, field applicable rapid diagnostic tests to identify patients with malaria, and artemisinin-containing medications that Karen has discussed. Here in the United States, we have approximately 1,500 cases of malaria each year. We see them at UVA. We have several cases a year. I was called about one just 2 weeks ago, and someone who had returned from East Africa. The victims are typically immigrants or travelers coming from Africa, India, Southeast Asia, and occasionally Latin America. We have excellent rapid diagnostic tests and effective treatments of malaria now in the United States. Our greatest challenge is to think of the diagnosis. So here's a take-home point for you. And you're all responsible now that no one that you know well dies of malaria in this country. The first is fever in the returning international traveler is malaria until proven otherwise. So you need to say this with me. So are you ready? No, I'm not kidding. Fever in the returning international traveler is malaria until proven otherwise. I've got to say it again because it's really important. Fever in the returning international traveler is malaria until proven otherwise. Point number 2, use medications to prevent malaria when you travel internationally to sites where it is transmitted. Think about that. Don't take a chance. We're very fortunate at UVA to have a wonderful travelers clinic staffed by Drs. Eric Hewlett, Tanya Thomas, and Shannon Muna, and it's a great source of information if you have questions. Today, the war on malaria continues on many fronts, led by giants here and abroad. At UVA, we are extremely fortunate to have Dr. Jennifer Guler, who's here today, Jenny, who has been studying the mechanisms of drug susceptibility and resistance in Plasmodium falciparum, which is the killer, and Dr. Kita Kita, who's developing new ways of rapid diagnosis. So in conclusion, let me thank Karen for an outstanding talk and her stimulating account of the Malaria Project in her book. Y'all need to read it. Looking into the future, I envision Karen returning to discuss a new book, which I think will be entitled Victory in the War on Malaria, the Rest of the Story. So Karen, I can't wait to read that book and hear your discussion. Thank you.
Thank you both so much. This is, again, a very rich and complicated story that moves in many different directions. We have a good number of minutes for your comments and questions. And we'll have a couple of mics available. If you have a question, please identify yourself before you ask your question or make your comment. But we look forward to a good conversation.
My name is Karen Knight, and I work in the Health Sciences Library. I thought your talk was incredible, and you're such a good storyteller. My father was a medical entomologist straight out of his PhD program going into the war and was in the first malaria control unit embedded in a combat zone. So I grew up hearing these stories, but you've provided such a rich, much more complete picture of all.
What was his name?
Kenneth Knight. He was part of the Skeeter Beaters. There's a book about the Skeeter Beaters. But, you know, I grew up knowing that malaria killed more soldiers than the Japanese at the time, is what they always said. So it makes me just really appreciate the whole story. Thank you so much. Definitely share your book with my brothers.
Oh, great. Thank you. Yeah.
Hi, I'm a retired physician. I'm 92 years old. I was a technologist in New Guinea for a year, malaria technologist. I went to school thinking I was going to get a 6-month course in malaria as a pharmacist mate, and they sent me out with a microscope and a rifle.
Could have been you
after
under that
6
tent.
weeks. And your pictures were fascinating. That's where I was. And I was attached to a naval CB unit. So a year in Dutch New Guinea, and then another year in the Philippines. But this was 43, 44, 45. My kids know the story.
Yeah, you know, I came away from this story a little biased because there were all kinds of political reasons for wanting to say that one thing or another brought the malaria rates down. But really by 1944, those malaria rates were tumbling, especially in the Pacific. And I think it comes through in my book that I was very biased towards the malaria units because I think they did. I think they made the biggest difference. It wasn't the Adabrin. They figured out how to use Adabrin, and it helped. But I really think it was the malaria units.
It was really okay to be yellow, and we were
Right.
all yellow.
Yeah. So thank you. Yeah.
Hi, I'm Justin Butter. I'm a fellow in geriatrics here and a wannabe historian. And I wanted to to go back to what you've been saying here at the end about this tension, and I'm going to give it a sort of category here between vertical approaches and horizontal approaches to eradicating or controlling disease, because I think it's a tension that we still very much live with today. And I wanted to just see what your comments would be about that. I think there are the bioethics of human experimentation, very powerful in what you've uncovered here in this story, but also the bioethics of how we distribute resources and think about how we use those very important and scarce resources in controlling and preventing disease. So I wonder if are there any sort of key other take-home points in that debate that inform what we're doing today?
Well, from my perspective, what you know informs me. I actually thought about the bioethics of making these choices on where the resources go, but they're so real, right? So when Ebola was exploding in West Africa, I was asked to write a piece for Time magazine that linked malaria. What can we learn from malaria to inform what we do about Ebola? And in that context, I really felt strongly that the lesson coming out of this book that I learned is the lesson that everybody needs to take from it, and that is, if you don't have basic health care on the ground, if you don't have basic health services on the ground, we will see again and again. It might not be Ebola next time. We probably will see another Ebola outbreak, but it might be something even scarier. And there's very little we can do to tamp it down if we don't have trained people on the ground in these countries identifying, isolating, and preventing the spread of the diseases. So for me, prevention has to be the answer. The drugs, I don't criticize the Gates Foundation or any of the pharmaceutical companies who have created nonprofit arms so that they can work on these drugs. And the vaccine that may or may not come out, probably not going to come out. I mean, all that is great work, and I think our scientists learn so much from that money here. But I do wonder what would happen on the ground if the balance was shifted a little or a lot, and we saw more health, basic health services created and sustained in places like Liberia. We should try it. Like we've been on this drug trail since the war. Really, the war shifted that paradigm. Before the war, we got rid of malaria through economics. The U.S. South outgrew malaria. The dams created electricity. Electricity brought industry. Industry created jobs that got people out of shacks and into homes with closed eaves, and they could afford screens, and those who could afford fans could blow the mosquitoes away, and those who could afford air conditioning could keep the mosquitoes out. I mean, it was this confluence of things, but it was economic development. We outgrew malaria, and that's pretty much a story of every country that has eradicated malaria. They economically outgrew it. So while I appreciate all the hard work that goes into making pills and vaccines, I really feel like trying a different course it might produce different results, longer term results. I mean, the reduction in infection rates is temporary, right? We pull out of there and those rates go right back up like they did in 1970 after the World Health Organization pulled out of their eradication efforts. Sri Lanka and India's malaria rates got down to close to zero. And when resistance spread and we could no longer use the drugs, and we could use DDT, which was another product of World War II, mosquitoes developed resistance to that. We recoiled, the West recoiled, the World Health Organization pulled out, and those rates went up even higher in India, and the death rates were higher because people had lost their acquired immunity to it. I don't know if that answers your question, but I feel, I'm not an expert, so I kind of quietly feel that way. I feel like it's, people who are on the ground really doing the work need to come to that moment where they decide what's better.
I have several thoughts. I think that we need to pay great attention to the injustices, social and economic, around the world that underlie many of these diseases, including malaria. I also think it's not an either or, it's a both and. We have various tools in our hands. Some of them now are mucins, which are for this moment effective, but likely they'll lose their effectiveness in time, so that clock is ticking. We tried DDT, and the mosquitoes became resistant, and we did great environmental damage. We now are thinking about genetically altered mosquitoes. And we have models of healthcare like Paul Farmer's outreach for HIV that need to be expanded into a more comprehensive community healthcare system. So I think it's both and we need to use all of these opportunities as quickly as we can to get our fingers around the problem.
We're pretty much out of time, but I'd like to mention last week's program was on mosquitoes and Zika, and certainly some of these same points came up about the challenges, not only on the front of trying to find a vaccine or something like that, but simply of the mosquito control in a lot of areas that those mosquitoes can breed in water in a bottle cap, and the difficulties of working in challenging environments to do that.
Zika mosquitoes were most plentiful and are most plentiful in the really poor areas. So this is it's another economic issue. There's been some talk of it in that context, but it is an economic disease.
Yeah, very much, very much. So again, thank you both for these programs. Actually, next week, we do not have a medical center hour. We've had to cancel because of unforeseen circumstances with the speaker who was to have come. So no medical center hour on October 26th. The next program is Wednesday, the 2nd of November, highlighting ethics consultation at UVA, resources for negotiating tight spaces. And we'll have 5 folks here from clinicians talking about ethics consultation here at UVA. And we'll hope to see you then. Thank you all so much. Thanks to Karen Masterson and Dick Pearson.